Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Jason Shepard. He's part of the University of Utah. He's a biochemistry adjunct associate professor, also in neurobiology and anatomy and ophthalmology. So we're going to talk about a concept I'll let him describe about how networks of cells work together and possibly talk to each other. So, Jason, thanks for coming.
Yeah, no worries. Thanks for having me.
Well, can you describe your research? What are you working on?
Yeah, so the main question my lab is trying to address is, how does the brain store information? How does the brain allow us to have memory or memories, and for up to a lifetime, despite the fact that most of the proteins in your cells don't last very long? And so we're trying to understand at the molecular level how the cells in your brain are able to store information and allow us to recall those experiences that we have throughout a lifetime.
Do you think it's inside of a particular cell is where things are stored, or what about the particular connections amongst, let's say, a bunch of brain neurons? That structure, maybe that retains somehow certain memories and things.
The basic idea is that there's a structure in the brain called the hippocampus that we know is absolutely required for making new memories, and we know a fair amount of how that structure is arranged, how the cells in that part of the brain act together. And when you are learning and you're having an experience, there's a certain specific set of cells in the hippocampus that are active. And it's those cells that are encoding that experience. And then after the experience, there's this consolidation period where those active neurons become connected to each other through their synaptic connections. And so that process is called plasticity. So synaptic plasticity, where the connections between those cells that were active during that experience become wired together. And so then when you recall the memory at some other later point, it's that specific circuit that's activated, and you only need a few cells in that circuit to activate the rest of the cells. And so we think then that it's this plasticity process of wiring up those cells into that specific circuit is how you get information stored. And so we're quite interested in the genes and proteins that are required for setting up the plasticity and allowing that plasticity occur.
Well, what's the fewest number of observed neurons that can store a memory or a response? Maybe in rats or in people. What's been observed?
The fewest number of cells. Yeah, I guess that's hard to say. Obviously, in animals that are not as complex, so like a fly, for example, which, all animals learn. So all brains have evolved to learn and adapt to their environments. In a fly only has about 1 million cells and in its brain, and maybe only a couple of hundred are required for a specific memory. As you move up into more complex animals that have, higher cognition, then it gets harder to say what the number is. And certainly in humans, when you have these rich, what we call episodic memories, where you have all these sensory information that is combined with your conscious experience, it's undoubtedly thousands to millions of cells that are actually required for that experience.
Well, the reason why I ask is perhaps that might be an easier model to understand. If there was an instance in a worm or a fruit fly or something or a cockroach, maybe only 10 cells are involved in something, avoiding light, something like that. Maybe
so for sure
quantify what's going on.
The history of this field sort of came out of classic experiments done by Eric Kandel, who won the Nobel Prize for his work. And actually, he decided to use a very simple system in the Plesia, which is a sea slug. And part of that reason for that was that the sea slug has these well-defined neurons, doesn't have a lot of them, and they're huge, so you can actually see them without a fancy microscope. And so his lab figured out a lot of the rules of plasticity using that simple model and simple forms of learning like habituation. So many animals have a reflex. If you touch them, they'll withdraw, an appendage. But if you keep on touching it, it learns that it's probably not dangerous, and so that it's habituated to that sensory information. So it's a very simple kind of learning in a very simple organism, but indeed that's how quite a lot of the initial rules about how these synaptic connections work came from. My lab, we work mostly on mice, but we also do some fly work, some Drosophila work.
So what have you been able to ascertain? I would guess there's a structural component certainly to memory and brain, but you say you're looking at a molecular level. What are you seeing?
Yeah, so my lab's kind of concentrated on this one gene called ARC. And in fact, this gene has taken us down an interesting line of work. Initially, I was quite interested in it because we know that if you take it out of mice, the brain is wired up normally, but they just cannot remember anything for long periods of time. So this conversion of short-term to long-term memory requires this gene. And so we've been trying to figure out why it's so important. We know that when you learn, this gene gets turned on. So it's rapidly made in those active neurons that make up the memory circuit. And then one of its functions is to regulate the strength of those synaptic connections between cells. And that kind of makes sense with the model. But a few years ago, we made this really interesting discovery that the protein that this gene encodes has this viral origin. So it's kind of an interesting story. And the bottom line is that this gene, we think, evolved from an ancient viral element that infected an ancestor 400 million years ago. And it's retained, what's really fascinating is it's retained a lot of the biology of this virus, and especially in terms of how retroviruses work. And we think that in our own brains, this gene is being made, it's retained some of this viral biology, where it's actually signaling from one cell to another cell using something called a capsid, so a protein capsid that viruses usually use to transfer their own genetic material. So kind of a wild story, and we're still trying to figure out exactly why you need this kind of biology for memory, but it's taken us down this very different road where now we think that part of how we end up getting this sort of complex ability to remember things came from this random virus, ages ago.
What does that mean? Does that mean that neurons are sending extracellular vesicles to each other as part of communication?
Exactly. So, what viruses normally do is they take advantage of host cell biology, and viral capsids, the final infectious particle, looks very much like an extracellular vesicle. But here we've got this endogenous gene that's being expressed in neurons, and it's getting out of cells in a specialized extracellular vesicle, where we think that this capsid that it's forming is transferring important information from one cell to another cell in the form of RNA. But we're still trying to figure out what that signal is and why you would need this. And it sort of doesn't fit in the classic model of memory, but we think it's opening new avenues for research in terms of really trying to figure out what's going on there.
I mean, from my interviews, talking to people, I mean, all cells put out extracellular vesicles in mass. They take them in too pretty readily. So how would you identify that there's a specialized EV coming out of the neurons? I mean, EVs, I guess, are notoriously hard to process, and they're tiny, and their membranes can shred easily in centrifugation. So How do you know what's in the EVs and how they're packaged?
That's a good question. I mean, it's a real problem in that field in general, and we also come across that. So, there's functional experiments. We've tried various ways of purifying these vesicles, and so you're absolutely right that neurons, just like other cells, are making a bunch of different extracellular vesicles, and the challenge is to isolate the ones that you're mostly interested in. And so ARC is only in a subset of like 10% of the vesicles. We've worked out some ways of isolating that population. We've also been using recombinant protein that we purify from bacteria that form the capsids, and then sort of do very reduced preparations to get at what the capsid can do. But we're approaching this from various angles, and one of the big questions, of course, is what is the cargo that ARC is actually transferring into the vesicle, and what's the functional relevance of that in the cells that take up those vesicles?
Can you manually establish connections between cells, or only the organism can do that? I'm sure you could break them selectively.
Manually.
How about establishing?
What do you mean?
Let's step back. So what do the connections look like when you have a bunch of neurons that, people would say are wired together? What does that mean? Physically, what happens?
Yeah, so in a neural circuit, you've got hundreds of cells, thousands of cells, thousands of connections. And so to sort of give you an idea of the size that we're talking about, the cell body is, a tenth of a millimeter at best. So it's 100 microns. And then these processes can go for millimeters. So they send those dendrites and axons out to connect to each other. But one well-connected cell can connect to thousands of other cells. So it's in an actual brain tissue, it's extremely densely connected. There's hundreds of thousands of synapses. So it's very difficult to try and manually manipulate them in a way. But what we can do is interfere with proteins that we know are important for either the structure of the synapse or the receptors at synapses, so we can target those receptors that are involved in the strengthening or weakening those synapses. And then we can also try and figure out what proteins are selectively useful for the plasticity of those synapses, not just the basal function of them. So there's definitely tools we can use, genetic tools and pharmacology we can use to interfere and look at how those connections work. There's also newer technology where we can actually put in channels that are light sensitive but are not normally active until we shine a light on them. And that's actually another neat way to causally activate a set of cells and their connections between each other.
Oh, you can add in additional connections that are light activated?
We can put in a light activated channel in a subpopulation of neurons. We haven't got quite to the point where we can put them in selective synapses, but that's actually something we're working on as well. But we can certainly engineer them so that they go to specific cells. So, for example, we can tag the neurons that are active during learning, come back later and shine a light on the cells that were tagged with the channel during learning, and basically reactivate that circuit, and then observe the mouse to see if the mouse seems to be recalling the memory. And that is indeed the case, that we can sort of come back and the animal's not in a environment. Let's say we conditioned the animal to know that there's a reward in a particular kind of context or a box or something. We could basically then substitute the box with light. We can shine a light on the animal's brain, and because we've tagged those neurons that are active during learning, now the mouse thinks that it's in that box where the reward is.
I mean, for instance, can you have a mouse run a maze that it has never been run through? It knows it already?
Yeah, we haven't got that far yet. So in order to be able to do that, we would have to know exactly which synaptic connections to activate and whatnot. And that we can't do that without the animal experiencing something or learning something first. So that kind of inception of memories, we're not there yet.
Well, if you could see the detail, find enough detail on what happens during a learning experience, I mean, you probably wouldn't be able to recreate it nearly exactly, but I wonder if you could make a few connections to that mimics it, and then maybe jumpstart the learning process somehow.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
No, that's possible. I mean, in a crude way, we can predict which neurons would elicit that kind of response. But in terms of, as you say, the detailed information in a particular memory, that we're far away from in terms of reconstructing without any prior experience.
I'm sure there's people that look at the connections in the network, and I would guess it's 3D in all directions. I mean, it's certainly not, I guess, 2D. But has anyone tried to picture it and see what they can elucidate from the structure of the connections?
Yeah, I mean, but we're talking about a scale that's immense, right? So one synapse is maybe 100 nanometers at best. And if you've got hundreds of thousands of these, you basically, the only way to reconstruct the full, what we would call connectome, is by electron microscopy. So there has been some recent, large-scale studies to try and reconstruct a certain volume of brain tissue, like a cubic millimeter. But the amount of information and data in that one cubic millimeter is like more than most of the computers that you have online right now. So trying to make sense of that information is what's challenging, even if you've overcome the hurdle of actually imaging that volume, that piece of volume. But we're, I think the pace of technology is pretty amazing. And so again, they've sort of done this with the fly brain, where they've reconstructed almost the whole fly brain to that kind of resolution where you could look at synapse level. But it's taken them years to reconstruct one fly, one brain. And so we're really far from figuring out how the dynamics work. So it's a snapshot of how the brain looks when you killed the animal. But the one thing that we have learned about how information is encoded and stored is that it's very dynamic, that there's new synapses, there's synapses that are getting removed, there's new connectivity. So it's an extremely hard problem, because you're now talking about trying to reconstruct these thousands to millions of synapses over across time, so that we have not been able to do yet.
Oh, so you say synapses get removed as well?
Yeah, you can think of it as sort of a sculpture, or a tree that you're trying to sculpt into a shape. And so you let certain branches grow, but you cull others, you prune basically the leaves of some of the branches. So it's the sort of very fine-tuned sculpting of the circuit that includes removing synapses, removing connections that don't make sense.
So, I guess I can imagine like a whole bunch of people in a room very close together, and they could reach out their arms and touch one another or pull their arm away, and they could change the dynamics of how everyone's connected, just as a really simple thought experiment. And I guess that would kind of approximate what's going on with neurons.
Yeah, kind of. So I guess the one way to think about it is that, so when your brain is developing and wiring up, it's the beginning. It's all about the genetics, and there's essentially cues that tell the synapses where to go. And then once you've got sort of that rough plan of how the brain works, then you need the fine tuning, sculpting, and that's where learning comes in and experience comes in and refines those connections and those synapses. And the way that can happen is in various ways. So we were talking about strengthening and weakening of synapses, and that can be at a structural level, but it could also be at the functional level, where you just have more receptors or more neurotransmitter that's being released.
So I guess what's the speed of signaling between cells that are connected by synapses versus if they're using extracellular vesicles? Like, if you look at the speed of action of those two, what's the difference?
It's big. So, when you're talking about electrical activity in the brain, the firing of cells, of neurons, that's in the millisecond domain. Whereas to move and make an extracellular vesicle and then to have it get taken up, we're talking about minutes to hours. So they're doing very different things.
I thought they were constantly being produced and taken in and remade, and I thought it was like kind of a continuous process. in varying degrees?
I mean, that may be the case for some extracellular vesicles, but in this case, we think it's a highly dynamic process that's controlled by this gene, and so it's timed to experience. So it's definitely not a constitutive process that's always ongoing. And but yeah, the time scale is quite different to the regular synaptic activity that you can measure.
So do you think the EVs are like a reinforcement for more important memories or learnings or long-term memories?
Yeah, exactly. So we think that perhaps this cell-to-cell signal is it's certainly involved in consolidation, and we know that takes minutes to hours to happen, and it's somehow stabilizing the circuit, and how it's doing that we don't know. So yeah, that's the sort of main question we're trying to figure out right now, actually.
Well, you could have a bunch of cells connected and see if you could block the EVs for a period of time and see what would happen to the network, maybe. If you had just a few in a dish that were connected, if you can keep them alive, and again, block the EVs somehow, would the connections fall apart, that kind of thing. Maybe you could figure out the interaction there.
Yeah, so we're approaching this in many different ways. So we have cells in a dish. We also have mice that we can interfere with this pathway. The nice thing about this looking like a retrovirus is that there's been decades of work on HIV and other retroviruses, and we have a structure of the capsid that we've obtained through our collaborators at the UK with John Briggs at the LMB. And so we can now predict which amino acids are required to make the capsid and then mutate them so that what we end up having is a mutant protein that cannot form the capsid, but it has everything else we would think normal. And so right now we're testing, whether those mutant mice or putting that mutant protein in mice, how it affects the network and whether it's absolutely required for that consolidation of memory process.
But when you say there's a capsid, do you mean like there's a membrane for the EV, and then inside of the EV there is RNA packaged inside its own capsid?
Exactly.
Wow, okay.
Yeah.
I know EVs are pretty similar to viruses. They can enter a cell and affect its function, et cetera. But so you're saying that, okay, some of these, our own genes are making virus-like particles. So
exactly.
Packaging.
We think actually that there's at least a dozen other predicted genes that have similar viral domains. And so we're also looking to see if those genes retain similar biology. And it's looking promising that there could be a whole repertoire of endogenous viral-like capsids that are being made in different cell types.
So there's no apparent instruction by a virus. These cells are not infected. They're just, they know how to make capsids in certain circumstances.
Yeah,
certain
exactly.
Genes know how to do that.
Yep. So these are real genes. They've lost all the other bits and pieces of viruses or retrotransposons, these transposable elements, and they've been cemented in the genome for many millions of years, and so they've become this sort of real gene. What's kind of cool is that this is an emerging theme that these transposable element sequences, these viral sequences, are all over the place in genomes. And the human genome has up to like 50% of our genome is of viral origin, and only 3 to 5% of the genome is actually coding for genes. And so it's turning out, I think, that a lot of these sequences can end up being used by evolution to create new genes.
So are these HERVs? Are these previously endogenized retroviruses that are forming these RNA and these capsids? Or is it, what you call, I guess, human genes that are doing this? Like this what is it?
These are just genes. They don't have, so they've lost those viral elements. So as you say, there's another group of sequences in the genome called endogenous retroviruses. And we were only just now sort of figuring out what's happening there. And it turns out, of course, that some of these endogenous retroviruses can actually be expressed, and especially in the context of aging and disease. But these are more recent events where, an ancient retrovirus, or not ancient, an older retrovirus has infected a population and it's integrated into that population. But it still has a lot of the viral bits and pieces that are that you can recognize, as opposed to this gene that has lost all those bits and pieces, and what has been retained is the structural component of the capsid and a few other things that make it look like a retrovirus, but it's not. So it's sort of, a little fuzzy in that we're making these distinctions between a gene, an endogenous retrovirus, a retrotransposon, and a retrovirus.
I wonder why there's this extra capsid packaging. Maybe if an EV gets ripped open, this will still survive. Maybe it's a more important cargo. Or maybe once the EV enters into a cell, the membrane gets absorbed into the cell membrane, and now you're left with the interior contents. And again, so that they can passage properly inside of a cell, they need their own capsid, some of them.
Right. Yeah, no, exactly. Good question. So we've obviously puzzled over this too. The extracellular vesicle field is lacked from mechanistic insight into, one, how do you select for cargo, and how do you actually get cargo into cells? The way viruses fuse membranes is they have a specific protein that can do that, and it's not clear what that protein equivalent is in extracellular vesicles. So the capsid, for one, as you say, can be used to stabilize cargo. So RNA is inherently very unstable, and there's lots of proteins and whatnot in cells that can degrade the RNA. One reason to have a capsid is that it's going to protect that RNA and have it in a very stable format, whereas a naked extracellular vesicle with RNA could really get rapidly degraded even once it's outside the cell. So that's one idea. The other idea is selection. So the capsid can certainly be a mechanism to select for specific cargo that can then get packaged into the extracellular vesicle.
Huh, that's crazy.
Yeah.
Have you been able to isolate these capsid enclosed, RNA fragments and study them, using electron microscopy and look at their structure and see if they have spike proteins or things like that on them?
We've started to do that. So we published a paper earlier this year in Nature Neuroscience where we reconstructed the fly capsid. And actually, if you look at the images on in that paper, there are spiky bits. There's spiky proteins. Spiky structures that are coming out of the capsid. But unlike a real retrovirus, this is all encoded by one protein, the GAG protein. And retroviruses, the spike protein that retroviruses encode is a separate gene that's in the sequence. So it's going to be interesting to see, what the parallels are here of what that spike protein is, but it's going to be slightly different.
Now that you've isolated it and looked at the capsid, does it look like any other viruses out there? Is it similar? And if so, to what kinds?
Yeah, it's very similar to HIV, for example, and foamy viruses. So retroviruses in general. Evolutionarily speaking, it's fascinating. So we actually think that it's most related to a retrotransposon, and so retrotransposons have most of the same bits and pieces that retroviruses have, except they don't have usually that spike protein, that envelope protein, but they are also able to form viral-like particles in a similar-looking capsid. And it's thought that the retrotransposons are ancestral to the retroviruses. So here we've got a brain gene that has homology and has evolved from an ancient retrotransposon that forms capsids that look like HIV capsids.
That's crazy.
Yeah.
I mean, that says that cells seem to have almost all living forms, I guess, seem to have a similar sets of abilities, and they can make similar things.
Evolution's very good at taking advantage of things that work. And so I guess it shouldn't be too surprising that if a virus can take advantage of host cell biology, that at some point the host can take advantage of the viral biology, because the viruses have solved some specific problems that would help the organism, and then evolution doesn't have to reinvent it. So, I think there's going to be more and more of this where we learn that some particular process in animals may have been already solved by another sort of more basic form of life. Another example of this is the evolution of the placenta in mammals. And that's a crazy story again, where evolutionary, the placenta has evolved a number of times, but in every time it has evolved, it's evolved because of a viral envelope protein that's allowed the placenta to fuse with the mom. So without that viral protein that has now become a gene called syncytin, we would not have placentas.
Yeah, I've heard some of the new COVID vaccines may express spike proteins and cause people to express antibodies against their own placenta if it's formed. So it could be a problem.
Yeah, I haven't heard that. There's definitely reports, or in many cases, where if a mom is infected and there's an immune response, that the antibodies to that virus can be transferred to the fetus.
Yeah, you just wouldn't want women to make antibodies against placenta formation and therefore be infertile. Recently, it looks like maybe a possibility, which would be horrible.
I think that seems unlikely with coronaviruses because that spike protein in the coronavirus looks very different to syncytin or any other placenta. I guess that's what I know anyway.
Well, hopefully so, yeah.
Yeah,
that's good, because you certainly wouldn't want it the other way.
Yeah, I think that I think if there's going to be issues down the road there, we would start to hear about them soon. But as far as I know, that is not something that we should worry about for COVID, at least.
I mean, there's a lot of things for you guys to figure out. So what are the next steps for your research? What's your goal? I mean, I guess like a 1 or 2 year goal. And what are some of the milestones you think that'll show you that you're on the right path?
Yeah, and I mean, it's we've got a lot to do, which is great, and we've recently got some funding for it, so that's also good. But so going back to this signaling role of the EVs that ARC is making, we obviously want to know, what exactly is ARC transporting? How do they get out of the cell? How do they get taken up into other cells? And then of course, what's the function? Do we really need this cell-to-cell signal for memory consolidation, or is it something else that we're just not aware of? And then we're also quite interested in the long run, figuring out what these other genes that look like they could also form capsids are doing. There's another family of them that are expressed in the brain, for example, and we don't really know much about what their function is at all. And so it could expand to sort of not just be one gene, ARC, but many other genes that could be important for extracellular vesicle-like signaling that use the same sort of capsid approach.
Well, again, since synaptic connections are super fast and EV communication is slow, again, do you think it's a reinforcement of important learned items to make them into more long-term memories or acquired skills? Or what role do you think the EVs have in bolstering this communication and memory and learning and all that?
Yeah, I would say that's where we're thinking that of course it's because it's slower, it's involved in somehow stabilizing the circuit. But until we know what the cargo is, we can't really, we don't really know. But let's say it's the main cargo is its own RNA, so it's making more of its own protein in these cells. One function of that protein when it's not forming a capsid is to remove receptors from synapses so that they become weaker. And so one idea is that you have these cells that are active that form the network for a specific memory, and then you don't want the surrounding neurons to be incorporated into that network unless they were active. And so you need a signal to those neurons to say, hey, let's become weaker, let's not interfere with that circuit. And so maybe that's what ARC is doing, is that it's able to go to the neighboring cells through these extracellular vesicles and weaken the synapses on them so that they're not ultimately actually incorporated into that circuit.
Are you able to tag something so that you'll see it expressed in EVs? And then if you look at a network of cells, you can see that the more EVs are being expressed in certain parts of a cluster or less. Or I was going to ask you about like with flies, if you've seen the structure, what do the endpoints of the network look like versus like the center? Is there a center? Are there clusters where control seems to be happening and ones that are kind of outliers? There's only a few connections to them.
No, exactly. So we're trying to tag the protein. We're trying to tag the cargo so that we could follow it in vivo in an attacked circuit. And that's the big challenge. So we can do some of this kind of imaging in dissociated cells in a dish, but that doesn't really recapitulate the circuit. So we're getting there. We think that it will be possible to tag cargo or protein so that we can then observe exactly where they're going when they're released, for example. And then in the fly system, in the fly brain, we're actually still trying to figure out if the fly arc gene is required for memory as well, because I glossed over this, but the mammalian gene that we're studying is in mammals, and it's basically in all land vertebrates, but fish don't have the gene. And so we think that this ancestral viral infection or transposition happened in between fish and amphibians. But then it doesn't make sense that a fly would have that gene, because of course the flies are invertebrates and are much older. So it turns out that the fly gene is actually a more recent evolutionary event that repurposed the same kind of retrotransposon or viral element, because other insects don't really have that ARC gene, or at least most insect lineages don't. So it's somewhat of a interesting, every time we've sort of looked at this, we start to determine various aspects of the story. It's just got crazier. But anyway, so the bottom line is that we're doing similar experiments in flies to see if they're involved, if the fly gene is involved in memory. We know that it's already expressed at synapses. We just don't know exactly which synapses it's expressed at.
Do you know of anyone looking to see if there's other levels of communication between cells, like biophotonics or any other ways? Or do you think these are the two ways that cells communicate? I mean, what else could there be?
There's many other ways. So there's not just vesicles being released, but hormones, which are basically naked proteins, are being released all the time. So there's lots of cell-to-cell signaling that happens through the release of naked proteins. There's also direct communication, where something called a nano tunneling tube, where essentially, as it sounds, the one cell.
Oh, TNTs, right?
Yeah. And then there's, I guess those are the main ones, where either the cell is releasing naked protein that can act on receptors, many cytokines, many hormones, that's how they act. And so the immune system is full of these signals where you've got proteins that are being released and acting on other cells. And so, and in terms of the extracellular vesicles, I think we're only just now figuring out some of those signaling functions in general.
Yeah, it's crazy. Just last question or two. What does biogenesis of these EVs look like? Can you see it? Do we know, like, where in a cell, these EVs are made and packaged and assembled and the capsid parts made?
Yeah, so that's another goal of ours. And it's somewhat of a needle in the haystack, because the size of these capsids are less than the light, the diffraction of light. So you kind of have to do EM to definitively show where they're forming. But the two possibilities are that they form at the cell surface and they bud out like an actual virus does, or they could actually form like a normal exosome, which has to go through this endosomal multivesicular body compartment, so membranes inside the cell, and then they get released as a bulk thing. So we're not sure yet exactly where they form and how they are released. So a lot of cell biology to still figure out.
Yeah, it's amazing. The more I learn, and like you said, the more you learn, it's like the complexity is just insane.
Yeah, I know.
What's the best way for people to find out more about your work and your research? Sounds like you get a lot going on. Where can they go?
I'm pretty active on social media. So Twitter, Jason Synaptic is my handle. We have a lab website, www.shepherdlab.org. And I actually had a TED Med talk that I talk about this work. So that video is, I think, accessible to most audiences, and you can find that on my website.
Well, very cool. Well, Jason, thanks for coming on the podcast, and it's been a great call. I really appreciate it.
Yeah, no worries. Thanks for having me.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.